in dilute and crowded solution, which is a very hot topic recently in the field.
Thanks so much, Yang, for the invitation to come here and also for your kind introduction. What I'd like to do is to tell you some of the work in our recent work in the lab, specifically on protein association. So we have been interested in this problem of protein association, that is the formation of a complex between a protein and other either macromolecular or small protein molecule targets. So we've been working on this for quite some time, and so in particular, we've been working on structure prediction for protein complexes, and also done work on analyzing the physical factors contributing to binding affinity. But today I'm going to focus on the kinetics of protein association, that is the calculation of rate constants of association between proteins. And much of this work has been done under dilute conditions, as many of studies of protein interactions are. But more recently, we've looked at what happens when the association occurs in a cellular environment. So many of you probably know that the cytoplasm of bacteria cells, and as well as inside organelles of eukaryotic cells, are highly concentrated with different kinds of macromolecules. So this is a David Gursell's rendition of the cytoplasm of E. coli. And the point of this picture is to show that the spacing between neighboring macromolecules, for example, between the ribosome and between neighboring ribosome, for example, is less than the size of a typical macromolecule. And so the question is then, does this crowding environment affects the biophysical properties of protein association, structure, equilibrium, and kinetic properties? And so I hope to have some time to tell you some of our recent work in this context. Now, most of you probably know that when a protein folds into a typically a tertiary structure, many of the non-polar or hydrophobic groups are buried in the interior, whereas polar and most of the charged groups are exposed on the surface. And the charged groups can either arranged in a way such that they form stabilizing pairs of opposite charges in some cases, but also in other cases, you may have a case where a patch of like charges is clustered so that it forms a binding site for a opposite charged protein, so that they can have electrostatic attraction between the protein molecules. And I'll show you some of those examples later on. Now, we now know that the proteins function in the cellular context, typically not in isolation, but rather through interactions with many other macromolecular and small molecule partners. And so just to make this point, for example, let's consider the example of protein synthesis. So we know that the synthesis of new proteins occur on the ribosome, involving the large and small subunits of the ribosome, the transfer RNA, as well as the messenger RNA, leading to the release of the new protein. But this is actually a highly simplified picture of what's going on. A more realistic picture involves many other protein factors. So the translation involves initiation, elongation, as well as release. And typically, the process of translation is really limited by the first step, that is the initiation. So if we look at the initiation process a little bit further, we now know that the initiation itself is really limited by the binding of the messenger RNA to the small subunit, and that process is helped by 3 initiation factors. I have 1, I have 2, I have 3, and part of the reason for the 3 initiation factors is to help speed up the binding of the initiation tRNA to the small subunit. So there is actually a problem here in that not only the initiation tRNA, but also all the elongation tRNAs can all bind to the same site, and and without the initiation factors, they all bind to the small subunit with more or less the same rate constant, and so therefore there is a high probability that you may have a wrong tRNA that's bound to the subunit, and that obviously can create problems with errors in protein translation. And so the problem is largely solved by the help of the 3 initiation factors. So with the 3 initiation factors, the rate constant of the cognate tRNA is increased by 3 orders of magnitude, so that it's basically a kinetic competition that's won with the help of the initiation factors. So the examples like this, specifically in this case, suggest that it is the distinct rate constants of association that really helps the efficiency and accuracy of the translation initiation process. And so there are this and many other examples suggest that often the biochemical processes are under kinetic as opposed to thermodynamic control, and therefore the study of the association process, both the rates as well as the mechanisms, are essential for understanding the physical basis of biological recognition. And so that is essentially the motivation for us to work on methods and experimental techniques to characterize the mechanisms and the rate constants of protein association. Now, if we look at the observed rate constants of protein association, we find that the observed rate constants span a very wide range, about 10 orders of magnitude. So there are complexes that are formed at rate constants that approach 10 to the 10th per mole per second. There are also complexes that are formed at rate constants that are around 1 per mole per second. So there is this 10 orders of magnitude difference. And so the long-term objective of our work is to develop computational methods that allow us to, for each given complex, to predict its association rate constant. And so I hope that I can convince you that we've now solved about half of the problem. So rate constants that are higher than 10 to the 4th per mole per second, and now we think we have a pretty reliable, robust, and accurate method for making rate constant calculations. Now, to understand this the computational method, we need to look into this process of association a little bit further. So for 2 proteins to associate to form a stereospecific complex, obviously the first thing they have to do is they have to come together, and not only they have to come together, they also by the time they come together, they also have to form have the relatively the right orientation as well as a close separation, and this such a intermediate along the pathway of association we refer to as a transient complex. And once they reach the transient complex, they have to undergo further conformational rearrangement and tightening to form the native complex. So simplicity, we can divide this whole process of association into then 2 sub-steps. The first is the process of reaching the transient complex And this is done by translational and rotational diffusion of the two subunits. That's followed by further conformational rearrangement to form the native complex. Now, potentially both subsets can be rate limiting, and we can capture the two steps in a simple kinetic model. So, in which we have the two subunits, protein A and protein B, they form this transient complex by diffusion. Now, once you form the transient complex, this complex can either dissociate to the two separate subunits, or can further undergo conformational rearrangement to form the native complex. So the overall rate constant of association is then determined by the rate constants of the three elemental steps. So in a this simple formula where the overall rate constant Ka again is determined by the three rate constants. Now, as I said, both substeps going forward can be potentially rate limiting. So if the second step is very fast, that is, you have fast conformational rearrangement, then the overall association will be rate limited by the first step, the diffusional encounter. And that is what's called the diffusion limited regime. Conversely, if the second step, the conformational rearrangement step, is slow, then the overall association will be rate limited by the second step. And so the problem that, the half of the problem that we think we have a solution falls in the first scenario where the conformational rearrangement is fast. That is the diffusion limited regime. So in this regime, we have developed a method that we refer to as the transient complex theory. And we can use this to predict both the rate constants as well as the mechanism of association. So the basis of this rate constant prediction is this simple analytical formula that we derived based on manipulating the diffusion equation that governs the association process leading to the diffusion process that leads to the formation of the transient complex. So in this formula, we have a prefactor, this Ka0. This refers to, we call this the basal rate constant. So this is the rate constant by which two subunits would find each other to reach the transient complex by pure random diffusion, that is, without the help of any external or intermolecular interaction. And then this process can be enhanced by the presence of long-range electrostatic attraction. And that help is captured by this Boltzmann factor, in which the energy is the electrostatic interaction energy in the transient complex. So to implement this method, then we need to do three things. So we first have to identify the transient complex. So roughly, this transient complex is located on the outer boundary of the native complex energy well. So that as soon as you reach the transient complex, that puts you at the border of the cliff into the native complex well, so that you can very quickly go down this energy funnel and reach the native complex. So we specifically we identify the transient complex by mapping the energy surface of interaction between the two subunits by treating the subunits as rigid, so that we only have three translational degrees of freedom and three rotational, relative rotational degrees of freedom. So we map the energy surface in this six-dimensional space, and by doing so, identify the transient complex. And then once that's done, we then calculate the basal rate constant, that is the rate constant by which the two proteins reach the transient complex by unbiased diffusion. And we do this by simply simulating the translational rotational Brownian motion of the subunits by doing what's called Brownian dynamics simulations. And these simulations can be relatively fast because we don't have to calculate forces that can bias the diffusion process. And then we capture the electrostatic contribution by calculating the electrostatic interacting energy within the transit complex. And then we do this by solving the so-called Poisson-Boltzmann equation. All right, so a bonus of this approach is that this formula teases out the two contributions of the association rate constant. So we separate the contribution of the basal rate constant, and this is dictated by diffusional process that's the unbiased diffusional process, and then the electrostatic contribution. So instead of in alternative approaches, you simply get an overall rate constant. Here, we can separate out the contributions so that we can gain a better physical insight into the factors that control the magnitudes of associated rate constants. And then I'll point out the benefit of this decomposition. So, and specifically, one benefit of this decomposition is apparent in our understanding of this wide spectrum. So very briefly, we can separate the whole spectrum into two halves: the diffusion control or diffusion limited regime, and the conformational change limited regime, and the boundary between the two regimes is of the order of about 10 to the fourth per mole per second for rate constant in about this size. And then in the diffusion limited regime, we can further separate into a region where the rate constant is of the order of 10 to the fourth, 10 to the sixth per mole per second, and that's the range of basal rate constants that we typically obtain. So those are the rate constants that you would obtain if two subunits associated without the help of non-range electrostatic attraction. So, for example, antibody protein, antigen and antibody binding typically fall in this range, and then we can infer that antibody maturation is probably not guided by maximizing association rate constant, but rather by optimizing PrEP affinity. Whereas there are cases where the rate constant, there are not many cases where the rate constant is greater than this basal rate constant, and that can only happen with the help of non-range electrostatic attraction. So whenever we have a rate constant that's 10 to the seventh per mole per second or higher, we can infer that these are systems where there's a strong electrostatic attraction, and that's characterized by opposite charges across the binding interface. So you would have one subunit that's a highly negative charge, and one subunit is a highly positive charge across the interface. And these surfaces represent the electrostatic potential of the two subunits. So red means a negative charge, and blue means positive charge. And so anytime we have a rate constant that's higher than 107 per mole per second, again, you can infer the presence of strong electrostatic attraction. And often we can argue why the charges are clustered in this way, because now you have to realize that clustering of light charges is actually not good for the stability of the subunits. So you can actually increase protein folding stability of the individual subunits by mutating some of the positive or negative charges. So therefore, there has to be some kind of biological reason for the clustering of charges on the two subunits. And often one can identify such biological reasons. At least one can propose such biological reasons. So take, for example, this well-studied system, Barnase and Barstar. This is an enzyme. This is a protein inhibitor of this enzyme. This is the enzyme that cleaves RNA, so it's a nuclease. So this pair of proteins are produced in a type of bacteria, and Barnase is produced as a cytotoxin. It's basically a protein toxin that's released by this bacterium to kill off competitors. So it's basically if you will, a weapon that's used to its own advantage. But if you think about it a bit more, then there is actually a problem here, because if it cleaves RNA, then the first RNA potentially this protein can see is the RNA of the producing cell. So therefore, the producing cell has got a problem of self-defense. So it has to have a very good self-defense mechanism for it to effectively use this weapon. And that comes in, the self-defense comes in the form of this of protein inhibitor, BASTAR. So in the producing cell, the two proteins are actually co-expressed, and then very quickly form a complex, and then it's the complex that's secreted to the outside, and then they dissociate and then gets imported so that bondage can do its job. So here is a case where it's not the affinity of the complex that's important, is the key here, but rather is the rate constant forming this complex, because you can have formed very high a complex with a very high affinity, but if the process is very slow, then some of the bondage molecules can escape and then target the RNA molecule of the producing cell, so that and the cell would die. So many other, similar reasons can be inferred for all these other systems, so this suggests that there are biological reasons for this high speed in association in these examples. So I want to now show you some examples. Yes? It's both. Yeah, because the long-range electrostatic attraction would both increase essentially speed up the process, but also pre-orient. Yes. Yes, it is screen. I'm sorry? Long-range, so yeah, I have to define what's long-range in our case is basically of the order of up to 10 angstroms. So basically the proteins sense each other at a distance of about 10 angstroms. Yeah, so that comes, if you, that's actually a very good question. So that comes from, if you look at this formula applies only for very stereospecific association. So if you have a non-specific binding, like two spheres touching each other, the effect of a non-range interaction is very minimal. It's only when you have very stereospecific interaction, then you have this Boltzmann factor. So basically, 3 orders of magnitude basically requires a free energy of about 2-3 kcal per mole. So that's how you can get this significant speed up. All right. So the first example I'm going to show you is the binding of a enzyme, a toxic enzyme that targets the whole ribosome. It binds to a specific site on the ribosome, in particular a loop of the ribosomal RNA, and once it forms this complex to this particular site, then the enzyme cleaves the RNA, and so that now the ribosome is inactivated, and then protein synthesis is stopped. So it's a highly toxic protein. So this protein is of the same class of something that you might have heard in the news. There is a in the past half a year or so, there was in the news that there was a guy that's sending ricin-laced letters to Obama and other people. So ricin has a protein that's harmonious to the system that we studied. Basically, it acts in a very similar way. It targets the same loop, and this loop is actually known as the sarsen-ricin loop. And the protein that we studied here is ricin rather than sarsen. Is sarsen rather than ricin. And part of the reason I want to show you this example is actually to make the point that our method is not limited by the size of the system, so we can study the whole ribosome, and so So we can basically effectively study any association process involving any macromolecular complexes. But a second reason I want to show you this example is that this is a system where the experimentally measured rate constant by a group at UCSF is the highest that's ever recorded, exceeding 10 to the 10, approaching 10 to the 11th per mole per second. And so the measured both the association of the ribotoxin to the intact ribosome as well as to the isolated ribosomal RNA fragment. And so the binding to the RNA fragment is in line with rate constants that measured with other protein-protein association. So this is of the order of about 10 to the 7th per mole per second, whereas the intact binding to the intact ribosome is now even higher than that by 4 orders of magnitude. So there is this additional 4 orders of magnitude boost from the presence of the ribosomal context. And the part of the reason we want to study this is so that we want to identify the factors that contribute to this additional 4 orders of magnitude rate enhancement by the ribosome. And so since, as I said, we can tease out the different contributions, then we can figure out the various factors that's contributing to this factor, and we find that half of this 4 orders of magnitude, about 100-fold, comes from the fact that you have this ribosomal context. So the ribosome is largely negatively charged because of the ribosome RNA, the phosphate groups on the backbone of the ribosome RNA, and the ribotoxin is positively charged. So they have this long-range, non-specific additional electrostatic attraction. But there is a second, the second effect that's more interesting and more subtle, and that has to do with the fact that the neighboring ribosomal proteins modifies the binding interface. So this is a representative configuration of the transient complex for the binding of the ribotoxin to the isolated ribosomal RNA fragment. Now, if you place this transient complex in the context of the ribosome, then one sees that you have clashes between the ribotoxin and the neighboring ribosomal proteins. So therefore, the transient complex has to be modified, and the modification is such that there is a rotation, so that a counterclockwise rotation, so that now you avoid the clash and have you can fit into class in neighboring ribosomal proteins. And this reorientation of the transient complex, what it does, it positions the positive charges on the ribotoxin very close to the phosphate groups on the ribosomal ribosomal RNA fragment. So that now the electrostatic attraction in the transient complex is further increased than what is in the in the case for the isolated ribosomal RNA fragment. So you have a concerted effort, both from the short range effect by modifying the binding interface, as well as the resulting electrostatic attraction that accounts for the additional 100-fold increase in the rate constant when you compare binding to the isolated ribosome fragment to the intact ribosome. So that's an example that demonstrates the importance of the electrostatic attraction as well as the binding interface, the shape of the binding interface. And I think those two factors probably account for, we're looking into how the initiation factors, the 3 initiation factors, increase the rate constants of the messenger RNA binding to the small subunit, the 3 orders of magnitude. I think so quantitatively we think that a combination of electrostatic effects as well as modification of binding interface can account for this kinds of difference. But again, we need to do quantitative calculations to establish that. So I want to now. Mm-hmm. Yeah, so that's you can describe the enhancement as a example surface diffusion. Yes, basically it's with the help of the electrostatic attraction, the two subunits basically stay longer, and by attaching to the neighboring surface, so you can model the process as a reduction in dimensionality. Yes. All right. So the second example I want to show is the binding of cytokines into cytokine receptors. So we got into this problem. So typically a cytokine has two binding sites, a high affinity one and low affinity one, and it binds to two receptor molecules, and so that it induces dimerization and the conformational change within the dimer that leads to activation of the receptor. And we got into this because we looked at experimental data for the binding rate constants of four different cytokines: erythropoietin, isoleucine 4, human growth hormone, and prolactin. They have very different biological functions, but the structures of the cytokines are similar. They're all Helix bundle proteins, and also the receptors are also highly homologous. And but the rate constants differ by about 4 orders of magnitude. So we were first of all curious as to what explains the binding rate constants, and also we wonder whether or not there are biological reasons for this difference. And so the structures, and this is the binding for the high binding rate constant for the high affinity site. So the since we have the structures for these complexes, and so we can easily do our transcom-based calculations, and we find that the basal rate constants are very, very similar. They differ basically by threefold, and it is really the electrostatic contribution that accounts for this difference. So the electrostatic interaction energy in the transit complex differ by about 5 kcal per mole, and that essentially accounts for the difference of this 4 orders of magnitude change. And if you actually look at the electrostatic surface, you can easily see why the rates can be so different. So in the highest rate is for EPO binding to its receptor. In this case, we find that EPO is highly positively charged, and the receptor is highly negatively charged. So you have strong electrostatic complementarity. In the other extreme, prolactin, we see that the prolactin does not have any strong electrostatic feature. Basically, it's a mixture of positive and negative, and similar for its receptor. And so simply looking at the electrostatic surface gives a physical explanation, and then the transcom calculation basically gives a quantitative explanation. So this again suggests that it's the electrostatic difference that accounts for the difference in observed rate constants. Now, and then the second question we wondered was why there was such a difference in rate constant of these 4 structurally related cytokines, and so we were coming up with I guess different kinds of reasons, but none of them was satisfactory. So we reasoned maybe since EPO was involved in producing blood cells, and therefore maybe you need fast association to produce enough blood cells, and then prolactin maybe you don't. And so finally we actually it's as I said, this it's not very satisfactory. And finally we looked at the circulation concentration, because we looked at because in biological processes you don't really directly care about rate constant. It really cares about the binding rate, and the binding rate is of course the product of the rate constant and the reactant concentration. So you can get the same binding rate by either having a high rate constant or by having a high protein concentration. And if we examine the circulation concentration of the 4 cytokines, we'll find that there is strong anti-correlation between rate constant and circulation concentration. So EPO with the highest rate constant is present at a very low, at a picomolar concentration, whereas prolactin with a low rate constant is present at 10 nanomolar concentration. So there is, so basically when you form the product, you basically find that this product is more or less constant. And this binding rate is essentially enough to, so that if you infer the half-life of this process, and this half-life is over the order of about an hour, and that's about the time that the half-life of the receptors before they are degraded, so that this rate constant is basically tuned so that it's fast enough just to escape before the receptor is degraded, and it's achieved by by compensatory changes of the rate constant and the circulation concentration. So therefore, the change of rate constants allows for the variation in the circulation concentration. That's basically the best explanation that we come up with. Now, since we calculate the rate constant, and in doing so also calculate the structure of the transient complex, and as I said, the transient complex is an on-pathway intermediate. So therefore, if we identify the transient complex and we simply draw a vector from the transient complex to the native complex, then we can infer the transition, the final transition from the transient complex to the native complex, so that we can infer a mechanistic model for receptor activation. So by calculating the structure of the transient complex and then doing this simple interpolation, we can come up with motions that are important for the final step of the receptor activation. So this is sort of a mechanism model that we developed based on the calculation of the transient complex. So you first bind the high affinity site, and then the low affinity site, and then in doing so, you have a relative rotation of the 2 subunits, and then the rotation then brings the 2. There are 2 kinase subunits that are attached to the intracellular domain, and this rotation results in the closing up of the 2 kinase molecules, so that allows the 2 kinase molecules to transphosphorylate, and the transphosphorylation then serves as the signal for activation of the receptor. All right, so we've now since we've done lots of tests of this method, and we've now actually automated the implementation of the transcom method and developed it into a web server. And so basically, it takes as input the structure of the native complex, and from that, then everything is the calculation is automated. You can basically enter your protein structure and then get the rate constant. I know Barrett's group has actually used this method quite a bit. We actually have now this web server up for now about 1.5 years, and we have a significant number of submissions to this web server. So if you're interested in testing this out, this is the URL for doing that. Just to advertise this method a bit, so this is a set of examples of that we've run through the web server. This is a set of 49 protein complex for which we found experimental values for the rate constants. They span 5 orders of magnitude, ranging from 10 to the 4th to 10 to the 9th and above per mole per second. And we fit in the structures of this of this complexes to the web server, and this is what comes out. And so the prediction is of the order of the accuracy of the order about 5 to 10-fold difference from the observed rate constants. And this complex is the span different biological functions as well as different structures, and now most of this complex are relatively rigid, but there are also cases where we can actually apply this method to flexible subunits. As an example, this is a case where one of the proteins is actually partially disordered in the unbound state, and this is example of uridine binding to thrombin, and so we adapt our method to the binding of disordered proteins, and we simply cut the disordered protein into fragments, and then we bind the fragments one at a time. And the fragment that gives the highest rate constant, we predict that as the overall association rate constant. And the reasoning for that is we think that the different fragments can bind to their respective sites in parallel pathways. Of course, the most contribution would come from the pathway that has the highest rate constant. And so we refer to such a type of binding as a dock and a coalesce to have this binding of the high rate fragment followed by the coalesce of the slow fragment. And so in this case, the disorder then allows the protein to escape from the sphere orientational restraints. If you were to bind this whole subunit as a rigid subunit, then the rate constant will be very, very low. So by having the fragments flexible and disordered, then you can have still have this kind of high rate constant. Now, so we've looked at this dock and coalesce mechanism and wonder whether what it applies to other disordered proteins, and we found that actually a number of other disordered proteins that have been studied experimentally also follow this dock and coalesce mechanism. So this is a system that's studied by Richard Kiraki's group, and what they found is that if you cut this into fragments, they find that one fragment, the N-terminal fragment, has the same rate constant as the full length protein, whereas the C-terminal fragment has a rate constant that's 3 orders of magnitude lower, and that is precisely what we predict using this our fragment-based calculation, transcom calculation. A different system that's studied by Peter Wright's group is the binding of PKIT to KICs, and here they actually proposed a model that's similar to dock and coalesce. They found that in the process they form the two subunits form intermediates in which one helix is formed first, and then the second helix formed subsequently. So we think that this the dock and coalesce mechanism is a general mechanism that governs the binding of disordered proteins in many cases. And we've now in addition to this system, we examined a dozen or so other systems using this fragment-based transcom calculations, and in all these cases we can explain experimental results quantitatively. So so we think again that suggests that this is a dock and coalesce is a general mechanism. Now there is a number of the study of disordered protein has become very popular, and there are a number of theories that have been proposed as to why disorder exists in proteins. And so some suggest it allows for promiscuous binding. Others suggest it's for allowing you can have a low affinity binding, and so we argue none of this seem to make too much sense. For example, promiscuous binding. In fact, now we know that's actually a bad thing, not a good thing. In fact, if you look at, there is a recent science paper in which the expression levels of proteins were examined, and they found that disordered proteins proportionally were expressed at a lower level, and their explanation was so that you can suppress precisely promiscuous binding. So you want, actually, that's something that you want to prevent as opposed to something that you want to promote with disorder. So so anyhow, we followed this fashion, and we came up with a new theory, and this is based on kinetics as opposed to thermodynamics. Now, and the reason that we formed this kinetic argument was because if you think about what functions typically disordered proteins fulfill, they typically are involved in regulation or signaling by forming complex with target proteins. Now, this kind of targets complex formation, in order for the IDP to work, of course, it has to be highly specific. As I said, promiscuity is actually a bad thing. At the same time, the complex has to be short-lived. Okay? If this complex has a very long lifetime, then basically you lose the switch-like function. Right? You're either constantly off or you're constantly on. So you have to, the complex has to fulfill three requirements: high specificity as well as short lifetime. Now, those two requirements, in a way, are contradictory to each other, because if you want a high specificity, how do you achieve high specificity? You can achieve high specificity either by very easily by having high affinity. Now, having high affinity means you either have high on rate or low off rate. All right. Now, the high on there is a limit to how much you can increase the on rate as limited by diffusion. So typically if there is without the help of electrostatic enhancement, you are limited to a rate constant about 26 per mole per second. And so if you want high affinity, let's say a nanomolar binder, then that means you have to have a dissociation rate constant of 10 to the minus 3, meaning that your half-life for the complex is about 1,000 seconds, and that would put you outside the range that's useful for signaling or regulation. So there is then now this dilemma, conflict between short lifetime and high specificity. Now, what happens if you make one of the subunits disordered? Okay, what happens is, because now the protein is disordered, that means that the disordered state is more stable than a hypothetical ordered state, right? And so therefore you lower the free energy of the unbound state, and by lowering the free energy of the unbound state, you increase the effective, the apparent dissociation constant. So now instead of a nanomolar binder, now you have a micromolar binder. And now with the same association rate constant, now your dissociation rate constant is only about 1 per second. So now that puts you in a half-life about 1 second. So that puts you in the range that's permissible for signaling or regulation. So we think that the disorder that ensures the association with its target is both highly specific, but also short-lived. And then we think that's a unique function of intrinsic disorder. All right, so I want to move on to briefly about what happens when association occurs in a crowded cell-like environment. So, as I said already earlier, that biological processes, protein synthesis, folding, binding, aggregation, they all occur in a context of a high concentration of bystander macromolecules. And the question is whether or not, or how much this crowded environment affects the various thermodynamic and kinetic properties Um, um, so now, potentially, this is a very complex problem, and we wanted to see whether we can still use simple theories to model such a process. Now, in the case of association in dilute solution, because the both species are present at low concentration, so therefore we can use a pair-based approach to model the association process. Now, when we have high concentrations of crowders, then this becomes a complex many-body problem. So the question is, can we still use a pair-based approach to model this process? And so we examine this question, and then we think there is actually hope that maybe you can still use a pair-based approach because typically when we have macromorphic crowding, the concentrations of individual species, for example, the rectin protein species, are still at a low concentration. It is the aggregate concentration, the total concentration of macromolecules is high, but individual species is still low. So as far as the rectin species are concerned, they are still at a low concentration. So if we can somehow model the effects of the crowders in a mean field type of way, we can maybe still use a pair-based approach. So we examined this question some years ago by using a very simple system. So we simply have a box of hot spheres that are doing their Brownian motion, and then the spheres are otherwise identical. We simply label one of them as our protein A, a few others are protein B, and then the rest as crowders. And then we, whenever A sees a B, then we assume that they form a complex and the system dies. So we basically monitor the lifetimes of the systems. We repeat this many times. We look at the lifetimes, and then from there we calculate the survival probability. And then we'll compare that with a pair-based theory. And this is a comparison between the simulation results and the pair-based theory prediction. And we find that the theory actually works out pretty well. And what you need to do is to account for the effect of the crowders. And there are two kinds of effects that we accounted for. The first is the fact that when you have crowders, then the crowders induce an effective interaction between the rectin molecules. So we introduce then the effective interaction. A second effect is that the diffusion of the random diffusion of the rectin molecules is slowed down by the presence of the crowders, so they have a harder time to find each other. So now these two effects are, in terms of effects, they actually oppose each other. So the slowing down of diffusion would slow down the association process, but the effective interaction effectively you induce attraction between the rectin molecules. So therefore, that would speed up the association. So those two effects could potentially cancel each other. You need to a modest net effect. And so anyhow, we can account for those effects by modifying our transient complex theory. So now the Bayes rate constant will be modified because, as I said, the diffusion is slowed down. So we simply introduce a new diffusion constant that's slowed down by the presence of the crowder. And then in addition, we introduce a effective interaction that's induced by the crowders. And so in this way, we can quantitatively account for the effects of crowders. And so now to calculate the crowder-induced interaction, we proposed what we refer to as a post-processing approach. I'll give you just very briefly the basic idea. So you can study experimentally the binding in dilute solution, and you can study the binding in a crowded solution. And then typically to get the crowding effect, you basically calculate the difference in the binding free energy, for example. So that's what you can do experimentally. Now in computation, we don't follow the horizontal path, but rather we follow the vertical path to, as explained shortly, this will avoid expensive calculations. So we simply take the unbound state and transfer it to the crowded solution and calculate it to transfer free energy. And we do that for the bound complex as well. And then if we take the free energy difference of the two vertical paths, then we would end up with the same free energy difference. So, and we refer to this as post-processing because we simulate the unbound state, the bound state, and then post-process the simulations by transferring them to a crowded solution. And this is something. So once we do the simulation, then we transfer it to the crowded solution, and each time we'll calculate the interaction free energy from that by averaging the different the transfer to different sites in the crowded solution. We get the transfer free energy. And so we applied this calculation to a system that's studied experimentally by Gideon Schreib's group, and they found, so they measured the binding of two proteins, TAM and a BLIP. It's a protein inhibitor to form a complex, either in buffer or in the presence of PEG or dextrin as crowding agents. They found that the observed rate constants really don't differ significantly among the different systems. Now we know that the presence of PEG and dextrin is going to reduce the diffusion constant of the two proteins by about 5-fold. So if diffusion, the reduction in diffusion constant is the only effect, then we would expect this process, the association rate constant will decrease by 5-fold. But because the crowding also induces an effective attraction, so that compensated for the effect of the slowing down in diffusion. So the net effect then is nearly null, and that explains what happens, what's observed experimentally. Now that is not to say all the effects of crowding is always null. So if we look at, for example, binary affinity, then there is no compensation from diffusion because this is an equilibrium property, and we do predict a increase in binary affinity, and this is a comparison of our theoretical prediction as well as with our own experimental measurements for the binding of two subunits of the DNA polymerase I complex. So there is enhancement by dextrin as a crowding agent, and the amount of enhancement in binary affinity is different depending on different species of dextrin. So a higher molecular weight dextrin has a smaller effect relative to a lower molecular weight dextrin, for example. So I think I want to wrap up with a few concluding remarks. I've showed you that we've developed this transient complex theory that we think provides a practical solution to half of the association problem, the half that has rate constant that's greater than 10 to the force per mole per second, and we're working now the remaining half that involves a conformational change in the association process. Diffusion, regardless of whether in what regime you fall, it always sets an upper bound on the association rate constant, and this bound is of the order of about 10 to the 6 per mole per second. But electrostatic attraction can bump up this rate by 3 orders of magnitude, so that you now have a very wide range of rate constants, and we've seen that this wide range allows proteins to fulfill its various biological functions. And finally, I've also showed you briefly that we've also studied computation and experimentally association under crowded conditions, and so our current efforts are devoted at more realistically mimicking the in vivo conditions. So with that, I want to acknowledge the people who did the work, the NIH for support, and thank you for your attention.
Yeah, with respect to your last point, that you're simulating, it is basically the counterintuitive idea is that they can find each other with some kind of magic attraction potential. But isn't it much more reasonable to say is that they're just in a cage And so once they have found each other, is then they can't, when they diffuse apart again, is they will reassociate. It's because they can get away from each other. Isn't that the explanation, a better explanation than a magical attraction force?
Oh, the cage effect is not going to work because what matters is when you have two guys that are separated, they have to find each other in the first place.
Right.
Okay? So the kind of process that we're interested in, these are diffusion limited. So what the slow step is really finding each other. It's not how long they sit together so that they don't separate.
No, but once they separate, is they can recombine again when they are in a cage. It's very reminiscent of what happens in a biocore machine. This is where things diffuse into the layer, and you would say if you diffuse further down the layer, the slower the association is, but that's slower, but the rebinding is faster as well, and therefore the kinetics is not affected. Very counterintuitive.
No, so the a cage effect would slow down dissociation, will not speed up association. Again, because association is determined by the process that bring you together.
Right,
bring the two molecules together.
But they rebind. They rebind. It is a rebinding.
The rebind still is the same thing. The rebinding still need to go through this diffusion process.
No, you just can't get away because the other molecules are around you.
So a very, very simple way to test your idea is to introduce a short range attraction. And in our model, and we've done calculations. If we have a short range attraction, that's not going to have a significant effect on the association.
Well, let me ask the question, what is this magic force then, which makes them to be attracted?
So it's not a magic force. Okay? So you think it this way. So if you have two subunits, okay, compare two situations, one, they are very far apart. The other, they are bound in a complex. Okay? In a crowded solution, the two guys that are separate apart would have a larger excluded volume. Whereas when you have a complex, you have a smaller excluded volume. So therefore, the complex is more comfortable in a crowded solution. So it's not a magic force. This is a well-known. This is in some circuits called a depletion force. So this is not some kind of magic force. We can actually calculate this force. So it's not some magic.
Okay.
Wouldn't you expect that in when you model your diffusion that it is also affected when you have these highly charged specific interactions, that somehow diffusion is changed, and when a highly charged molecule diffuses through solution, that it's actually slowed down due to its high charge?
So the presence of macromolecules can affect the electrostatic attraction in different ways. And a simple way this effect can happen is when you have the dianeutral constant of your solution is changed. So a highly concentrated protein solution will have a lower effective dianeutral constant relative to water. So therefore, the attraction, because of the decrease in the dianeutral constant, the attraction is enhanced. But at the same time, the charges on the bystander proteins can also contribute to the effective ionic strengths. So that can also, so you have this sort of counteracting contributions.
I'm saying that because there is recently been a simulation of crowded protein cytoplasm, crowded cytoplasm. I think they showed when they take into account these electrostatic interactions, then they can achieve like the same order of magnitude diffusion coefficient inside the cytoplasm as opposed to when they don't do it.
Right. Yeah, I think that's the understanding the diffusion constant in a crowded environment. That's a very active area of research. I don't. To get it right is actually very, very difficult.
Quick question. Have you examined the? Oh, thank you. Whoa, it's loud. Have you examined the effects of like physiological ionic concentrations in the cell and the macromolecular, like the crowding effect?
Yeah. So I mean, if you mean by physiological ionic strengths, meaning 150 millimolar, the answer is yes. We actually usually start a wide range of ionic strengths. Okay. Yeah. For example, the binding of the ribotoxin to ribosome, we went all the way from I think about 10 millimolar all the way to about 700 millimolar. So we usually span a wide range of ionic strengths.
So I guess I'm sort of returning to my previous question and Eric's question. So the range of the depletion interaction is the size of the depletants.
Right.
So again, it's not enough to speed up the center of mass diffusion by tons, right?
No, so remember
So
the answer to your earlier question about 10 angstroms. So our
Yeah.
Crowders, the size is of that same about 10, 20. So it is long-range in the sense of of 10 angstroms.
Great, but so if it's speeding up your reaction rate, it's not doing so by getting their center of masses close to each other faster, because that effect is linear in the radius of the capture radius, and you can't get a big effect that way. Right.
So again, so the effect comes because you have stereo-specific binding. So it's true if you simply have two uniformly reacting spheres, you're not going to get this kind of effect.
Right. But then I think that's exactly the cage Eric was talking about. That what's going on is that it's keeping the centers of mass near each other longer so that you can find the right orientations to bind. Is that accurate?
That's one way of. I don't disagree with that.
Okay.
So, but to say that that's the only thing is simply preventing from dissociation. That's probably not accurate. Yeah. So I think you can.
I see. Okay. Yeah. Okay.
So when you make your calculations for binding affinities, you're starting from the crystal structure. Do you know how much, like, if you made small deviations to the crystal structure, does it affect your calculation?
So our calculation, what I showed is for a delta G. That is the effect of crowding on the binding free energy, on the binding free affinity. So we avoid by precisely, we know calculating the absolute binding affinity is a hard problem. So we don't follow that's why we avoid following the horizontal path, because that's what we would have to do. We follow the vertical path so that we simply look at the effect of crowding on the initial state, on the final state. Okay. So so for both, I think the question is more random. If the question is directed at if we calculate rate constants using docked or homogeneous model structures, how accurate it is, and we've done actually a number of cases like that. In fact, the example of the ribotoxin bound to the ribosome, that's actually a homogeneous based model. So so we have used homogeneous models and docked models for rate constant calculation. Yes.
So the related question is how reliable is a transient complex boundary?
Yeah. So we've examined a few cases of that sort. So there was a recent case where we used homogeneous model to get the transient complex, and then we found very recently there is a new structure came out. So we did a new calculation. So the Bayes rate constant we found is differ by about twofold, and then the electrostatic contribution is very, very similar. Precisely because part of the reason it's robust because in the transient complex the two subunits are separated by about a layer of solvent. So it's not very sensitive to the precise positioning of the side chains.
Transition from that transient complex.
Yeah, so I think that will be very sensitive to the structure of the final complex. Yes.
Okay, great. And we. Okay, last question.
I have a small question.
Okay.
What is the basis for
So there are different definitions of disorder. I think one definition is where operationally you can determine whether or not a protein is disordered by simply measuring, for example, secondary structure. If there was no secondary structure, then one can imagine there was no tertiary structure. So there are different ways of defining whether or not a protein is disordered, and usually this experimentally is fairly straightforward. You simply use using make a second dichroism measurement and find there's no second structure.
Great. I'm happy to see that this great talk generated a lot of discussion. But we are slightly run out of the time, so we will stop here. And let's thank you again for Huan-Xiang's really wonderful talk.
Thank you.